back, students, for today's lecture. Let me start by introducing myself to you all. My name is Shazeen Khan, and I'm from Department of Botany, KTHM College, Nasik. Today, we are here to discuss the topic of integration of metabolism from the chapter of Metabolomics, subject Biochemistry for MSc Part First, credit for semester second. In the last lecture, we have discussed what is metabolomics, what is the concept of metabolomics, and we have seen the different types of metabolism which takes place in organisms. In today's lecture, we will be seeing the topic of integration of metabolism, key junctions in metabolism, and the different pathways of integration of metabolism. You can see in front of you one diagram depicting the integration of metabolism. In this chart, there are various pathways are depicted here, which appear to be very minute because these are all the different pathways which operate in the living organisms for the synthesis or breakdown of different products. They are interconnected with one another at some key points, and that only we are going to study today. That is integration of metabolism. This is one chart depicting how the different various biochemical reactions they are interconnected to one another. Now metabolism, it is a continuous process with thousands of reactions occurring simultaneously in the living cells. All the metabolic pathways are integrated with one another. Now there are some key junctions in the metabolic pathways. As we said that the different metabolic pathways they are interconnected, so there can be some key junctions which will be common, or there can be some common points from where one pathway it is connected to another pathway. So there are three important compounds which act as key junctions in metabolic pathway. The first one is glucose 6 phosphate, second compound is acetyl CoA, and the third compound is pyruvate. So these three molecules they act as key junctions in metabolic pathways. Depending on the body's condition or depending on the body's energy condition, energy levels, these compounds they can convert itself into different pathways for the production or degradation of biochemical products. You can see here the first key junction of integration of metabolism, which is glucose 6 phosphate. We can see that glucose it is phosphorylated and it enters the cell. It cannot be reconverted to glucose by most of the cells. It forms glucose 6 phosphate. So whenever energy is abundant, ATP is abundant, glycogen synthesis is favored. So glucose 6 phosphate, it forms glucose 1 phosphate, which then goes into the pathway of glycogen synthesis, which is the storage form of carbohydrate in animals. When ATP is low, or when the body's energy levels are low, that time glycolysis is favored. So the glucose 6 phosphate compound, it is converted to fructose 6 phosphate, which then forms pyruvate, and it enters the Krebs cycle to release energy. Then whenever NADPH is in low amounts, pentose phosphate pathway is favored. That time glucose 6 phosphate it is converted to 6 phosphogluconate, which forms ribose 5 phosphate, and hence the pentose phosphate pathway is favored at this condition. So pentose phosphate pathway is favored during this condition, and by the pentose phosphate pathway, different reducing equivalents are synthesized, and these reducing equivalents in turn are required for different pathways of the organisms. Now the other two key junction in integration of metabolism are depicted on the screen. The second compound which we have seen from the list, which is pyruvate, and the last one is acetyl CoA. Pyruvate in anaerobic condition, it can form lactate. Also, pyruvate can be degraded to form amino acids. Pyruvate can enter the Krebs cycle to form oxaloacetate. When energy is in low condition, oxaloacetate can again form glucose 6 phosphate from the process of gluconeogenesis. This pyruvate can go from the linking step to Krebs cycle to form acetyl CoA. This acetyl CoA can be useful for the synthesis of fatty acids. This acetyl CoA can be completely what you call oxidized or broken down to release CO2. This acetyl CoA can also be used for the production of cholesterol and ketone bodies. And the intermediate in that pathway is 3-hydroxy 3-methyl glutaryl CoA. This is also one schematic diagram representing the major synthetic and degradation pathways, how they are interconnected to each other. You can see that glucose, it can be converted to glucose 6 phosphate. Then depending on the energy requirement of the body and the levels of reducing equivalents, it can undergo or it can be changed into various compounds. When energy is low, glucose 6 phosphate, it undergoes glycolysis to form pyruvate. This pyruvate can form acetyl CoA, which can enter the Krebs cycle. Also, when energy is in excess amount, this glucose 6 phosphate can form glycogen, which is the storage form of carbohydrate. Acetyl CoA can be used for the synthesis of fatty acids, which in turn form triacyl glycerols. In the During the time of low energy, these fatty acids, they can be broken down by the process of beta oxidation to release acetyl CoA and NADH2 and FADH2. And all these processes you can see in this schematic diagram. Now let us see what are the different steps in metabolic pathways. First one is glycolysis, then is fatty acids oxidation, then is degradation of amino acids. Fourth one is citric acid cycle. Next is oxidative phosphorylation. Next one is hexose monophosphate shunt. The next step in metabolic pathway is gluconeogenesis. And the last one is glycogen metabolism. We will see in detail, we will see in brief what are these different steps. First one is glycolysis. Now, glycolysis, it is the degradation of glucose to pyruvate or the degradation of glucose to lactate. In anaerobic condition, it is lactate. The glucose, it degrades to pyruvate to generate 8 molecules of ATP. This pyruvate is then converted to acetyl CoA. Next step is fatty acid oxidation. Fatty acids, they undergo sequential degradation with the release of a 2-carbon fragment, which is known as acetyl CoA. Whenever one molecule of acetyl CoA is released, the energy is trapped in the form of NADH and FADH2. Next step is degradation of amino acids. So amino acids, when they are particularly consumed in excess amounts, then required for the protein synthesis, so they will be degraded to meet the fuel demands of the body. The glucogenic amino acids, they can serve as precursors for the synthesis of glucose, and the ketogenic amino acids, they are the precursors for acetyl CoA. Next step is citric acid cycle. Acetyl CoA is the key and common metabolite produced from different fuel sources. For example, carbohydrates, lipids, amino acids. Acetyl CoA enters the citric acid cycle, also known as the Krebs cycle, and it gets oxidized to carbon dioxide. The citric acid cycle, it is a common metabolic pathway for the oxidation of all foodstuffs. Most of the energy is trapped in the form of NADH and FADH2. Next step is oxidative phosphorylation. The NADH and FADH2 molecules produced in different metabolic pathways are finally oxidized in the electron transport chain, also known as ETC. This electron transport chain is coupled with oxidative phosphorylation to produce ATP. Next step is hexose monophosphate shunt. This pathway is primarily concerned with the liberation of NADPH and ribose sugar. This NADPH is actually a reducing equivalent. It is utilized for biosynthesis of several compounds, including fatty acids. And ribose, it is an essential component of nucleotides and nucleic acids. Next step is gluconeogenesis. The synthesis of glucose from non-carbohydrate sources is known as gluconeogenesis. Several compounds, for example, pyruvate, glycerol, amino acids, they can serve as precursors for gluconeogenesis. Glycogen metabolism. Glycogen is the storage form of glucose, mostly found in liver and muscles in animals. It is degraded and synthesized by independent pathways. That is the glycogen metabolism. So let us see the different organs which are involved in the metabolic pathways. First is liver. Next is the adipose tissue. Third one is the skeletal muscles. And the fourth one is brain. So we will be seeing how different metabolic pathways it occurs in these four organs. First, you can see in this table. In this table, we can see that what are the energy compounds which are utilized by the different organs and which energy compounds are exported from these organs. So you can see liver, it preferably utilizes amino acids, glucose and fatty acids, and it exports glucose, fatty acids and ketone bodies out of it. Adipose tissue, it only uses fatty acids and it exports out fatty acids and glycerol. The skeletal muscle also, it utilizes fatty acids along with glucose and it only exports out lactate. Now brain, it utilizes only glucose and ketone bodies also it utilizes, but it is only during the starvation condition. Otherwise it only uptakes glucose. And for the export of energy, brain, it does not export out any biochemical compound. So the metabolism, it can be studied under two conditions. First is metabolism in a well-fed state, or also known as the energy-rich stage. The second state, it is starvation. Metabolism in starvation, also known as energy-deficient stage. So we will be seeing how the different organs of which are seen in the metabolism, how the different pathways are operated in these two stages, and how the integration of metabolism takes place in these two conditions, that is the energy rich stage and energy deficient stage. Now you can see the diagrammatic representation here representing metabolism in energy rich stage. We can see the four organs we have which we have previously mentioned here. First is the liver, adipose tissue, muscle and brain. So we are talking up here about metabolism in energy rich stage. During the energy rich stage, that is well fed stage, when energy is in high amounts, it is indicated by the enzyme insulin. Glucose, which is obtained from the food material which we eat, it is transported from the different transporters present in the intestine and body, and it is uptaken by blood. So from the bloodstream, these different organisms, it uptakes glucose. So we will start with one organ at a time. Firstly, we will start with liver. Now you can see glucose is uptaken by the liver, and since it is a energy rich stage, that time anabolism is favored. That is, synthesis of new compounds will be favored during energy rich stage. So glucose is uptaken from the bloodstream in the liver, and since it is in high amounts, so glucose, it will be converted to glycogen, which is a storage form of energy. This glucose can also be converted to pyruvate, and pyruvate in turn forms acetyl CoA. This acetyl CoA can be utilized for the synthesis of fatty acids, and when fatty acids are formed, they will in turn form the triacyl glycerol, which is a storage form of fats. These triacyl glycerols, they can then be transported to the muscles or to the adipose tissue. There also, these fatty acids, they can either be converted into triacyl glycerols or they can be broken down into acetyl CoA, which can be utilized for various purposes. Also, you can see that glucose, it is also uptaken by adipose tissue and the skeletal muscles, which will be utilized to produce pyruvate and glycerol, and hence the Krebs cycle operates in the adipose tissue and also the skeletal muscles, so that different processes can take place properly. Also, brain, it only uses glucose, so you can also see there is uptake of glucose in the brain, and in the brain also, glucose, it is converted to pyruvate. Pyruvate, it is converted to acetyl CoA, and Krebs cycle is operated in this manner. So you can see that in the energy rich stage, glucose uptake is favored, and synthesis of compounds, different new compounds are synthesized, or excess amount of energy, it can be converted into those compounds which can be stored, which can be used as storage form of compounds or storage form of energies. For example, glycogen in the case of carbohydrates, triacylglycerols in the case of fats, and proteins in case of amino acids. Now, next condition which we are seeing here, it is the metabolism in energy deficient stage, or you can say a starvation stage. In this time, anabolism is not favored. Catabolism is favored. That means whatever the storage form of products are there in the organs, they will be broken down so to release energy. During this stage, the uptake of glucose is limited. Why? Because brain, it only uses glucose. So glucose uptake by different other organs, it is reduced significantly. Whatever glycogen, suppose if we start with the liver, whatever glycogen is present in the liver, it is broken down to glucose. This glucose, it is released so that it can be utilized by the brain to continue its functioning. In the liver, you can see glycogen, it is broken down to glucose. This glucose is converted to pyruvate, pyruvate to acetyl CoA, and acetyl CoA can also be converted to ketone bodies. Now, ketone bodies, they are a group of compounds which can be utilized by the brain if for the next 2 to 3 days body does not get any energy. So this ketone body, presence of ketone body, it is actually a marker enzyme to indicate that the energy level is very low in the body, and during starvation for more than 2 to 3 days, it is present in the body, which is the ketone body. Now you can see that glycogen, it is broken down into glucose. Then also in the adipose tissue and in the skeletal muscles, you can see, let us start with adipose tissue first. In the adipose tissue, fats are deposited. So these fats, it will be broken down into fatty acids and glycerol, and then again the fatty acids and glycerol also, they will be further broken down into simpler products so that energy is released. In the muscles, you can see in the skeletal muscles, the different proteins, they are broken down into amino acids. Then the amino acids, they can be transported to the liver so that from the amino acids, by the process of gluconeogenesis, there can be production of glucose. And glucose, as I have said earlier, that glucose uptake by different organs, it is limited. Whatever energy, whatever storage compounds are broken down into glucose, that glucose, it is transported to the brain, because in brain there is a blood brain barrier is present, and only glucose can be transported, or it can be uptaken from the body by the brain. So glucose, it will be sent to the brain. If the body does not get any energy for more 2 to 3 days, and the reserve glucose, it is exhausted, that time, only in that conditions, the brain, it will be functioning on ketone bodies. So today's student assignment is write in brief about different types of metabolism with examples, and you can send it on the given mail ID. Summary of today's session. Today we saw how the different metabolic pathways operating in an organism, they are interconnected with one another. They are interconnected with the help of key junction points which are present, and those key junction points were glucose 6 phosphate, acetyl CoA, and pyruvate. We saw that depending on the levels of energy in the body, these key compounds, they can lead into different pathways. Also, we saw how the different pathways, first, we saw the different pathways of metabolism and how they are interconnected with one another during two stages, that is, an energy rich stage and an energy deficient stage. And we also saw how the different organs in an animal, that is, the liver, adipose tissue, skeletal muscle, and brain, the different pathways operating in these four organs are interconnected with one another. In the next lecture, we will be seeing the regulation of metabolism, how different enzymes act, and how in cell signaling it leads to the regulation of the different enzymes of metabolism. Thank you. Thank you so much, dear students, for watching today's lecture. We will meet next time. Thank you.